Guys, my name is Karina Garcia, and today I will explain the disease cycle of the bacterial canker of tomato caused by Clavibacter Michiganensis. This is a seed-borne disease, and in my opinion, it's very interesting. So, let's go to the next slide. Here in this slide, I have two pictures. This first picture I got from an article, and this second picture I got from a book. So if you're interested about this disease, you can check these citations here in the bottom. Okay, let's start with the third slide. In this slide, I put the picture of the disease cycle, and also I already have my diagram with the titles, and also I placed some letters to try to explain how I was looking for the information, because this picture is not like as the disease cycle that are usually um, fungi disease cycle, because now we are talking about a bacteria. Okay, let's start. Let's start with the survival, how this bacteria survives. If we check here in the picture, we can see that here mentions that the bacteria, well, survival in soil plant debris. So this bacteria survives in soil plant debris. So next, let's go with the primary inoculum. Uh, about the primary inoculum, I had to look for more information because I couldn't see some picture here mentioning what was the primary inoculum. So after I read that information in the articles, I realized that the bacteria in the soil is the primary inoculum. This bacteria could be in the plant debris or also could be in the soil. But these bacteria have more advantage, like more time, can survive more time if this bacteria is in plant debris and not alone in the soil. Next, the dispersal. The dissemination of this bacteria is by splash of water from contaminated soil and also running. Um, this information also is not mentioning here. Like I had to look around here because here also is dissemination through pruning, but it's like a little bit confused because we were talking about bacteria in the soil and now they are mentioned dissemination through pruning. So, but I got the information and I wrote here. This information about a splash of water from contaminated soil I got from an article because I couldn't find here. Okay, let's continue with the infection cohort. This bacteria is gonna enter or is gonna infect the host through wounds in roots, stems, or leaves. I also got this information through roots or wounds, through stomata, guttation droplets, and other natural openings, to finally arrive to the vascular system. And here's a picture of the vascular system. Talking about the colonization, once the bacteria is in the vascular system, the spread in the xylem vessels is gonna start. And then the bacteria can go to the surrounding tissues. And when I talk about surrounding tissues, it's gonna go probably to the leaf, to the stems, maybe, and also can go to the fruit and finally arrive to the seeds. Okay, and why that part is important? Like to know that the bacteria is gonna move to the stems, to the leaf, or it's gonna move to the seed or the fruits, because these like bacterium seeds, bacterium plants are gonna be our secondary inoculum. For example, if I'm a grower or I'm a research and I need to extract seeds of my plants to sow those seeds and to do other works, if my seeds have the bacteria, I have a very high percentage of possibility that when my plant grows, it's gonna show the symptoms of the disease. And of course, I'm gonna ruin my plants of my new research. And also, bacteria in plants. Why is very important this? Because if we talk about the tomato, some activities or some cultural activities in tomato is the pruning. A cultural activity in tomato is the pruning. So if we're doing this activity in a plant that is infected, we could use, a scissor. We can start pruning this plant, and then we can move to another plant that is not infected, and we could start moving the bacteria from an infected plant to a health plant. So that's one way we can start moving the bacteria. And of course, also using seeds, infected seeds. And something that also is very interesting for me is about the bacteria survives in soil plant debris. Sometimes, well, we're gonna see like plants already dying because the symptoms, for example, if the bacteria is in the vascular system, probably the plant is gonna die because the plant cannot get water, cannot get minerals. And finally, if this plant is just, if the grower leaves the plant in the soil, this plant is gonna be, they had a, it's gonna be like dry, very dry, and some parts of the leaf are gonna stay in the soil. And the bacteria take advantage of that can use part the tissues of this plant to just stay in the soil and survive. And the cycle is going to start again. But also, we can spread the bacteria by the dissemination through the pruning. If we do not infect a scissor or any tool that we are using to do the pruning, we can easily spread the bacteria from infected plants to healthy plants. And also, if we get these seeds infected with the bacteria, we can start sowing these bacteria without knowing that the bacteria is already there, and we can think that are healthy seeds, and then we can start doing again the pruning, and we are going to infect all of our plants. So this bacteria is very interesting for me because I have that mechanism that stay in the seeds. Well, guys, I hope you could understand my explanation, and here is the bibliography if you need it. Thank you for your attention.